Okay, so thank you for introduction. So in this talk, I present our decade-long collaborative research to develop biomarker for mental disorder using resting state fMRI and machine learning. And our key focus is generalizability, ensuring the biomarkers work reliably across multiple sites. And the mental disorder characterized by the clinically significant disturbance in cognition, emotion, and behavior. And now they are becoming one of the most serious social issues, with the number of patients continuing to rise. And by 2030, so mental disorders are expected to be leading contributor to healthy life years loss. However, the current diagnostic system DSM-5 relies on a self-reported questionnaire and symptom-based interview is not effective for guiding the treatment. For example, 60% of patients with depression do not achieve remission with drug treatment. To address this issue, so research for new classification system based on the biological data such as gene, blood, and brain data conducted worldwide, expecting objective diagnosis, personalized treatment, and new therapies. And the brain biomarker can be broadly divided into the two categories, the structure and brain. And the structural biomarker measured with MRI, diffusion MRI, or CT provides static information about anatomy, and such as gray matter volume or cortical thickness or connectivity pathways. And these features are relatively stable over time and useful for chronic changes. In contrast, brain activity biomarkers come from fMRI, EEG, or MEG. They capture dynamic aspects of the brain function, including neuronal oscillation, functional connectivity, and task activation patterns. And unlike structure, activity fluctuates moment to moment, making it variable for identifying functional dysregulation. And our study focuses on this second type of markers, as functional dysregulation is a key character of mental disorder. And the resting state functional connectivity is a widely used tool for investigating the brain network and also discovering brain activity biomarkers. This method quantifies the extent of the coactivation in spontaneous brain activities, and the extensive research highlighted the individual, its relation to the individual characteristics and the brain states and anatomical connectivity and neuronal signals. And in experiment fMRI during rest, where subject is asked to be relaxed without thinking anything particular, is measured for 5 to 15 minutes. And the resting state fMRI signals appear random at first glance, but reproducible pattern emerge when we analyze them statistically over longer time window. And typically, the functional connectivity is defined as a correlation between two time series from different brain regions. For example, so red and green region, so is a functional connectivity between red and green region defined as a positive since their signal changing in the similar way, while connectivity in green or blue region is zero since they looked independently changing. And in this way, we can represent the connectivity patterns, whole brain connectivity patterns in the form of connectivity matrix by taking correlation between all the pairs of the brain regions. And the data-driven biomarker development using this resting state functional connectivity and machine learning is an emerging field in neuroscience. However, the multi-site generalizable biomarker development is challenging. According to the review paper in Nature Neuroscience 2017, So they defined the 4 levels of evidence for clinically practical biomarkers: development, prospective validation, generalization, population level. And achieving this generalization level where biomarker is generalizable to the multiple scanner and multiple lab is in particular difficult. At the time this paper was published, the 2 Alzheimer's biomarker had been reported above this chance level, this level, but no mental disorder biomarker has not reached this level yet. And in Japan, the 2 nation projects have been launched to develop the neuroimaging biomarker. Both share the common feature of a multi-disorder and large multi-sites and large-scale data collections. And in the former SRPBS project, they measured about 2,000 subjects, including patients with major depressive disorder, schizophrenia, autism spectrum disorder, bipolar, and so on, using unified imaging protocol. And followed BMV project, so they measured about more than 5,000 subjects with multi-disorder and using newly developed unified protocol based on a human connectome project standard. And by leveraging these large-scale data sets, so we have conducted the 2 lines of research. And the first line focuses on the strengthening the evidence level using conventional functional connectivity features. This line. And the second explores the new features derived from resting state fluctuation, aiming to improve the accuracy. Today, so in the following, I will present a series of studies from this first line of research. And this research is collaboration with many institutes from participating in the SRPBS project and the Brain Mind Beyond project. And the first question we asked was how much does the functional connectivity differ across sites? One approach to address this question is a traveling subject experiment. In this experimental paradigm, one subject visits the multiple sites, possibly different scanners, and measures his brain activity repeatedly at each location. And this experimental paradigm is very costly, but it gives unique opportunity to discriminate the biological factors and measurement factors in real human brains, revealing the site-specific measurement actually differences. And we conducted data analysis combined with traveling subject data to multi-disorder data set from a total about 1,200 runs. And we visualize the functional connectivity data of the 9 sites on the 2-dimensional space using principal component analysis. And you can see the significant bias in the one site, HUH here. And to quantify this site-specific measurement bias, so we conducted the linear fixed analysis to this 2 data set. And we have the measurement bias found to be the larger than the group difference between a mental disorder and healthy control, indicating the necessity to reduce this measurement bias. And using this linear model, so we subtracted this part. So site-specific bias is disappeared like this way. And then we trained and evaluated the MDD vs Hiroshi control classifier with relatively large number of samples, about 1,300 runs, using Grasser's multimodal parcellation. and also traveling subject harmonization method. In this analysis, so for discovery data set, we use about 700 runs from SRPBS project. So measured with unified protocol, but different scanners. And for independent variation, so we use about 600 runs from independent another project with heterogeneous protocols and also different scanners. And the developed biomarker classified with this independent validation data set without overfitting and without clear bias in each site. Left-hand side is discovery classification performance in the discovery data using nested cross-validation, which achieved balanced accuracy of 66% and AUC, area under curve of 0.74. And which is actually very similar value. It's obtained for independent validation data set. And this is particularly surprising from machine learning perspective, given the high dimensionality of features compared with the sample size, as well as the variability introduced by different scanners and different protocols. And among the 70,000 connections, we identified the 25 significantly and frequently selected by the RAS classifiers. And some of these connections were consistent with the previous studies, such as left-right intra-connectivity previously reported common to the multiple mental disorders, and the connection starting from subgenial ACC, therapeutic target to depression patient. And most of identified connections show the underconnectivity in MDD patients, while only a few, such as thalamomotor and audiovisual connections, show the overconnectivity for MDD patient. And we apply this developed biomarker to the patient with another disorder, so schizophrenia and autism spectrum disorder patient, to see the commonality between disorders. And as shown in this figure, so SCZ patient, colored green, is more similar to MDD patient, while ASD patient, colored purple, it's more like Hiroshi controlled, indicating this biomarker axis representing the biological factors, something common to the MDD and SCZ, but not ASD. And we later tested this marker on the new data acquired at Hiroshima University. And importantly, so this was a fully blind test. So YT in ATR labeled the distinct state functional connectivity and sent to the only label to the OG in Hiroshima University, and he checked the answers. And without any retraining or recalibration, the biomarker achieved accuracy comparable to that of the discovery data set, this accuracy. We also conducted a comprehensive evaluation of the biomarker development pipelines. And biomarker development involves 4 key processes: parcellation, functional connectivity, computation, and harmonization, and machine learning. Each has the multiple options. To identify the best performing pipeline, we tested a total 360 pipelines and evaluate them based on accuracy and between dataset stability. And okay, so we found the last classifier is actually near optimal, so not so much different from the best classifier in this pipeline research. Also, cluster parcellation and tangent correlation tend to have better performance And interestingly, among the top 8 of top pipelines, so connectivity weight patterns are very actually similar, common classifier weight patterns observed, suggesting the robustness of this feature in data set. And also using the largest traveling subject data set by combining BMB and SRPBS traveling subject data. So I precisely quantified the unwanted measurement bias, measurement variation and biological variations here, compared with the so disorder-related factor. And in a single connectivity level, actually this unwanted factor dominated the actually disorder-related effect, but by multivariate connectivity biomarker like RASO, so we dramatically reverse this relationship by this way. And I estimated so feature selection time by 3.9 SNR increasing, feature weighting by 2.9, and ensemble averaging by 1.3, and a total 15 times SNR improvement is obtained by this last classifier. Okay. Finally, I will talk about the functional connectivity biomarker for subtyping MDD patient. Subtyping here means grouping patients who share the similar biological factor. From a machine learning perspective, subtyping is an unsupervised learning problem and is much more challenging than supervised learning problem, in particular high dimension data. In the paper published in Nature Medicine in 2017, Drysdale et al. Identified the 4 subtypes in the latent space discovered by the canonical correlation analysis between the Hamdi score and the pre-selected functional connectivity. And they reported the one subtype has a higher RTMS responsiveness. But 2 years later, Dinger et al. Point out the instability of their method. And for example, only one removal of the one subject changed the result so much. And these studies show the clinical significance of the MDD patient subtyping, but robust subtyping has not achieved yet. And our strategy was to identify robust subtype of MDD patients that generalize across 2 independent data sets, purely in data-driven way. And we prepared 2 data sets, which were essentially the same as those used for diagnostic biomarker development. And because the functional connectivity has a very high dimensionality, and the feature reduction was essential, and we applied a 2-step approach, which we call the hierarchical supervised unsupervised method. And in the first step, we performed the functional connectivity pre-screening based on discriminability between MDD and Hiroshi control, and reducing the connectivity from 10,000 to the 100, typically. And in second unsupervised clustering step, so we use the multi-view co-clustering algorithm by Takeda, developed by Takeda et al. This is a new algorithm, is rather robust to the modestly the high dimensional data because of its capability of the partitioning features automatically. And we repeated this process across multiple pre-screened FC sets and evaluated the clustering robustness using the adjusted lambda index. And finally, after identifying the most robust clustering solution, so we evaluated the clinical significance using the drug-naive MDD patient who had received the pharmacological treatment, and comparing their symptom scores before and after treatment. And it is important to note that the fMRI data were collected at the week 0. So that means fMRI data is not known the result of the 6-week Hamdi scores. And most of this is a pipeline research exploration, and the most stable subtyping was obtained by 3 edges connecting the thalamus and postcentral gyrus shown here. And if average of the 3 connectivity and standard deviation of the connectivity, and each patient are plotted in this axis, so patient distributed this way, and clustering algorithm grouping patient with these colors. And basically, the mainly mainly subject clustering according to this average axis. And we evaluated the clinical scores for the subtype 1, which has a positive correlation over connectivity, and subtype 2 is the weak connectivity, and subtype 4 is under connectivity cluster, which has more than 10 subjects for statistical analysis. And for 0-week Hamdi scores, we did not observe significant difference. But for the 6-week Hamdi score, the subtype 2 shows significantly smaller than the subtype 1. And we observed a significant difference in Hamdi improvement rate and responsiveness rate and remission rate. So overall, so we found a robust subtyping which predict this drug responsiveness. As a control analysis, so we examined whether identified subtypes were associated with age, sex, and sites, but none of these factors showed a statistically significant relationship. As another control, we attempt a subtyping based solely on clinical symptom scores, so Hamdi scores. However, this approach did not predict antidepressant responsiveness. Okay, so now summary. So we did the data-driven biomarker development of mental disorder using resting state fMRI and machine learning. Particular focus on generalizability, the multi-site data, and starting from the understanding site difference by traveling subject data. So we successfully developed the FDD biomarker for objective diagnosis and some subtyping predicting responsiveness drug. And the same method is applied to the ASD biomarker, SCZ biomarker, and has also successful result in generalizability across sites. And the future direction, so first, so there are several future directions. So one is improving biomarkers. And this will require creating larger data set, maybe across much big project. Also integrating the multimodal data to characterize each individual from multiple perspectives. It's also important. Also, functional connectivity is rather modest reproducibility. So maybe another feature engineering will enhance the performance. And then feature engineering, representation learning, and foundation model is one possible approach. And to make biomarker clinically useful, we need a longitudinal study to distinguish traits from the state marker. Also, prospective treatment trials required. And finally, so to link the biomarker with treatment, it is necessary necessary to understand the biophysical mechanism of the biomarker alternation. And for example, so functional connectivity is a statistic over a long time window. We don't know actually what causes this causes the FC alternation. And this is the alternation of the thalamocortical connectivity observed in MDD patient. And the MDD patient show the overconnectivity and the motor and the temporal cortex, while underconnectivity in the frontal cortex. And the simple question here is what's the cause of this connectivity pattern alternation? To test whether, for example, this thalamic excitability could explain these findings. So we use the Wilson core model fitted with human connectome project data. And by systematically changing the thalamic excitability parameter, then observed corresponding alternation in the bold functional connectivity in simulation. And if we have the thalamus excitability is large, so over connectivity in the temporal and part of motor cortices and underconnected frontal cortices are reproduced. And of course, this is just a preliminary analysis, and it's not verified. No verification is still lacking, but looking ahead in the next 5 to 10 years, I expect the more studies which will combine the data-driven approach with biophysical modeling, ultimately advancing clinical translation. And this study was made possible through the contribution of many collaborators. I sincerely acknowledge the efforts, dedication to this series of the study. I also thank the funding support from the MEXT and AMEDO, Brain Mind Beyond and Brain Mind 2.0. Okay, that's all. So thank you for listening.
Yamashita, thank you very much for the comprehensive talk. So we have only 2 minutes for question and answer. We have now 2 questions from question and answer panel. One is it possible that in the insular connectivity, the claustrum is also included?
Sorry, I don't know the claustrum. What is a claustrum? Maybe second question. Let me start from second question.
Thank you for your talk. My question pertains to the methodology of your resting state fMRI studies. How do you instruct participants to achieve a true resting state without engaging in active or deep thought process? And how do you verify their compliance?
Yeah, basically, so instruction is just we give the instruction to the subject, and actually, and we can check the actually sleepiness score after experiment, but it's very hard to actually guarantee the level of the thought during the resting. So in that, it's some well debated some criticism of that resting state experiment part. And actually, we have modest reproducibility, so in resting state functional connectivity.
Thank you. Although there are more questions, unfortunately, it's time to go to next talk. So, Yamashita-san, could you answer this question after your talk? And
okay, so I input the answer to the question. Okay.
Yeah. Thank you again, Yamashita-san.
Thank you for the kind introduction. I'm Takufumi Yanagisawa from Osaka University. It's very my pleasure to present our work here. And today I will present our recent work on the dynamic mode decomposition (DMD) to extract the physiologically meaningful features from the neural signals and to apply it for the neural biomarkers and the decoding. First, And foremost, this research has been conducted by these outstanding and exceptional researchers. Professor Kawahara and Professor Yamashita are the expert in the DMD dynamic mode decomposition and also brain dynamics, as you may realize, and they lead this research. And additionally, Dr. Shiraishi and Majima and Fukuma, each young talented researchers apply this DMD to the our intracranial EEG and resting state EEG or cohort study. And they demonstrated the potential usability in the clinical setting. So today, I primarily discuss the content of these three papers, which represent their achievements. Okay, so here is today's outline, and I will start with the motivation and the limitations of the conventional spectral methods, and then I will introduce the DMD and show three studies decoding the information from the ECG and efficient and interpretable features of the DMD for the real-time application and biomarker discovery from the resting state EEG. And finally, I will discuss the future direction. Okay, so the central question of my talk is how can we extract the physiologically meaningful features from the neural signals? And this is the motivation behind the DMD. So for example, we record the ECG electrocortical graph signals by the electrodes on the surface of the human motor cortex. And while the subject performing these kind of hand movement, three types of hand movements, according to the sound cue. And recorded signal were evaluated around the movement onset for each channel, and to extract some neurophysiological features to relate to the movements, we apply the FFT or wavelet to the signals. And actually, for each electrode, we can calculate this kind of time frequency plot, and that shows an increase of the power at the high gamma range and the decrease of the power at the alpha and beta frequency range. And and also the spatial map of these frequency powers represent the localized high gamma activities on the motor cortex, and also the kind of distributed broad alpha beta power decreases on the sensory motor cortex. And as shown in this video, the gamma power changes its distribution depending on the movement types. That is, these spatial and temporal patterns characterize the movement information. Although such analysis demonstrated the neurophysiological features of ECG, FFT and wavelet have some limitations. First, they are usually high dimensional with redundancy because there's many channels and many frequency bands. And also they are applied for each channel independently so that their spatial integration is lacking. And lastly, they are difficult to handle non-stationary signals. And to overcome these limitations, we are interested in dynamic mode decomposition, or DMD. DMD is a data-driven method originally developed in flat dynamics to extract the coherent spatial-temporal patterns and applied broadly across the field, including the neuroscience. And actually, DMD shows the better better separation of the non-stationary spatial-temporal patterns compared to the PCA or ICA. And here, DMD decomposes the much varied time series into a dynamic mode, and which has the three key properties like a spatial pattern, intrinsic frequency, and the growth decay rate. And this framework allows us to represent complex neural signals in a compact and interpretable low-dimensional form. And formally, DMD assumes linear mapping between the consecutive time points at x and xt plus t delta t. And here, A is a transition matrix that governs the dynamics of the system. Then we apply the singular value decomposition to the data matrix with dimensional reduction. And finally, we can obtain the mode, frequency, and the gross decay rate to represent the dynamics of this X. And it should be noted that this gross and decay rate enable to represent the non-stationary signals. And also the frequency is determined from each mode based on the dataset itself. For example, we applied the DMD to the superimposed signals of the simulated data. So here there's two signals which distributed in space -10 to 10. There's the distribution like a bell shape, and the amplitude changes in the time, oscillatory time changes, and also the amplitude decay diverse in time. And we superimpose these two signals, make this one simulated data. And we apply the DMD to this simulated data and obtain at first we obtain the four singular values. And these four singular values corresponds to the each frequency and the mode. And actually, this mode is the complex value on the code in line with the spatial positions. And therefore, by multiplying the mode and the frequency oscillation, we can get the complex value, the oscillation in the real value and the complex values. But here, these two oscillations complex conjugate. Therefore, these two components disappeared by adding each other, and we can finally get the one real value, which is reconstructed from the simulated data. So, as you can see, the DMD can decompose the oscillation in the mode and the frequency with the decay properties. So it seems that DMD can extract the features of non-stationary spatiotemporal patterns. Then we have, but we have some questions to apply the DMD to neural decoding. First of all, how we can use these modes and frequencies to the decoding, and also whether DMD can improve the decoding accuracy compared to the FFT or WebMed. So when we apply neural decoding to the mode, we need to notice the difference between the FFT and DMD. In FFT, the neural decoding usually uses the power at some common frequencies. For example, we can pick up the power at some certain frequencies for each trial or movement and compare the value to classify the signals. On the other hand, in the DMD, the mode frequencies are different among trials, so because it derived from the dataset itself. For example, in this trial, 10.3 Hz and 20.1 Hz was obtained, but in another trial, 9.7 Hz and 20.5 Hz was obtained. So, and therefore, we cannot directly compare these two modes based on the same frequency values. Therefore, we use the projection kernel to classify the modes. The projection kernel measures the similarity between two subspaces by evaluating how much their directions overlap. Since this kernel compares the subspace, each mode does not need to reside in the same dimensions. So the projection kernel can, and also the this kernel can be easily computed by the multiplying the transposition of the. Then we calculate the gram matrix based on the projection kernel. Here, the ECOG signals was converted into the DMD modes. Then the projection kernel was used to build a gram matrix that represent the similarity between the trials. So trial by trial gram matrix was obtained. And we can apply, for example, the support vector machine to this gram matrix to classify the ECOG signals. And actually, we get the classification accuracy of about 80% using the DMD modes that was significantly higher than that using the FFT. Therefore, we demonstrate that DMD features improve the motor decoding accuracy compared to the FFT. This suggests that DMD captures spatiotemporal characteristics of the ECOG during the movements better than the FFT. But the problem remained here because we used the gram matrix. The use of gram matrix causes high computational cost so that the real-time decoding becomes difficult. When training the decoder, computational cost increases quadratically as the amount of data grows. Furthermore, the decoding newly acquired data requires recalculating the gram matrix, whole matrix each time, which necessitate substantial computation. So in addition to the computational cost, the gram matrix is difficult to interpret the feature of ECOG signals. Therefore, for the next challenge, we try to make an interpretable and fast features for the of the DMD that is used for the real-time calculation. Here, the Dr. Kei Majima actually found a beautiful relation between the projection kernel and the vectorized form of the representation. So the projection kernel can be rewritten as an inner product between the vectorized form of a function that corresponds to concatenated modes multiplied by H transpose. Here's the derivatives, but I cannot explain it well. So we can use this concatenated modes as a feature of the DMD for the neural decoding of the DMD modes. And we call this feature as the spatial DM features. It becomes the matrix of the number of the channels regardless of the number of modes. So for example, for the ECOG signals for the three types of the hand movements, the gram matrix become the spatial DM features of each trial, each class of the movement, representing the characteristic of the movement type. So as you can see, there is some high values of the diagonal part of the DM features, and it corresponds to the electrode numbers. So that means that some electrodes have the high value of the spatial DM features, and the changes of the value was observed among the classes, and that means these values representing the type of the movement. And actually, when we apply these features to classify the movement types, we can get the high classification accuracy using the DM features. And especially because the feature is the kind of vectorized form, we can apply some regularization term, something like a L1 norm, and to improve the accuracy, and we could get the higher accuracy than the conventional features. And also the time length spent for the training and test of the data set was significantly reduced using the proposed feature. Here's the proposed features. And it shows that we can use these DM features for the real-time online decoding, which is necessary for the brain-machine interface. So the spatial DM features achieved fast and accurate decoding, and actually this decoding is suitable for the real-time application, real-time BMR application. And lastly, we have tested whether we can use the frequency information to characterize the EEG. So to consider the frequency characteristics of the DMD, we created two types of signals here. The left one and the right one generated in different way, and both data consist of the 10 channels, channel 1 to 10, and for the channel 1 and 2, we added the signal of the 20 hertz and 10 hertz, and for other 10 channels, we added the signal of 20 hertz and noise. Okay, and in the left panel, left signals, the 10 hertz signals was continuously added. So this is the continuous case. On the other hand, the right panel, the 10 hertz signals sometimes disappear during the trial. And it observed among about the half of each epoch. And so there is some kind of switching behavior appears and disappears. And so this right example shows some kind of non-stationary state of the frequency characteristics. So we compare how these stationary and non-stationary behavior affect to the frequency estimation of the DMD. So we apply the DMD in each case, yielding a frequency around the 10 hertz and 20 hertz. And when determining the frequency distribution using all 4 components of the SVD, we could get this frequency histogram. They are quite similar between the stationary and non-stationary case. And also when we apply the power spectrum density calculation using the FFT, there's no significant difference between the stationary and non-stationary case. But when we calculate the frequency distribution using the 2 components of the SVD, there are significant difference between the non-stationary and stationary conditions because the reduced number of the component pick up the most dominant frequency among the each trial and makes it the difference between the stationary and non-stationary cases. And we hypothesize these non-stationary properties of the signal characterize the dynamics of the resting state of EEG of neurological diseases. And actually, we apply the DMD to evaluate the frequency histogram of the resting state EEG among the dementia patient. So this figure shows the healthy subject and Alzheimer patient and frontotemporal dementia patient. And for each representative subject, DMD was applied for 600 seconds of EEG data. And as you can see, there is a clear increase of the frequency around 10 hertz, and there is a peak of the histogram at the 10 hertz in healthy volunteers. On the other hand, this peak can become rather broad, blur in Alzheimer's disease, and the peak was shifted to the low frequency, but the PSD shows only a reduction of the power. And also in the frontotemporal dementia, the peak exists in 10 hertz, but another peak appears at the lower frequency range, but there is no peak in the PSD. So in this case, the PSD, the frequency histogram captured some difference of the diseases specifically. And actually, these are the average of the frequency histogram of 3 types of the patient. And as you can see, the histogram decreased at the alpha range and the beta range in AD and FTD. And also the frequency increases at the AD and FTD in the frequency range. And you can classify these AD and FTD using the histogram around these lower frequency range. But this significant difference was not observed in power spectrum density. Only we can see some difference between the healthy volunteer and the dementia patient. And in fact, when we apply the decoding to the frequency histogram, we can get the higher classification accuracy compared to the power or other spatial DM features. And okay, maybe we can skip. And finally, also we tested the same things for the Parkinson's disease. So this is the resting state EEG of the Parkinson's disease patient and the healthy volunteers. So there are the difference between the power spectrum density in these two conditions, but we can see also the difference of the frequency histogram between these two states, and we can classify Parkinson and the healthy better using the frequency histogram better than the power or other spatial DM features. So as a summary, temporal frequency dynamic of cap features, I mean the frequency histogram captures the intrinsic frequency distribution in the resting state EEG, and it outperforms the FFT for the classification of dementia and Parkinson's disease, and it provides the novel biomarkers beyond the conventional power spectrum. Okay. And finally, I'd like to briefly introduce a recent effort. As shown here, we can represent the brain activity as a combination of the multiple oscillations, and we are working to create a digital twin that mimics such activities. By applying this to epilepsy patient, we expect it can be used for the virtual epilepsy surgery to resect focus region and diagnose various diseases such as dementia. And specifically, we are aiming to develop the technology to create personalized digital twins by measuring the brain activity using the OPM, which is the next generation magnetoencephalography. And if you are interested in this kind of research direction, please feel free to reach out. Okay, this is our summary on the by efficiently capturing the spatiotemporal patterns of ECG, DMD improves the accuracy of neural decoding, and through the dimensionality reduction, DMD extracts the characteristic frequency features and then enables the discovery of new resting state EEG biomarkers for dementia and Parkinson's diseases. And combining DMD with novel measurement technologies such as OPM and digital twin models, we allow more precise representation, prediction, and control brain activity. So this work was supported by many collaborators and funding, and thank you for your kind attention.
Yanagisawa sensei, thank you for the very interesting talk. Now we have 5 minutes for question and answer. Do you have any question or comments? A question in Japanese is also welcome. So, may I have a ask question? You show that the frequency of DMD, your method is superior to the power spectrum. Why your method is better than? Why method can capture the difference between the conditions?
Yeah, thank you so much. Yeah, that's a very interesting point. And we are thinking that the DMD is much better to capture non-stationary changes of the frequency component. So, for example, if we so in this case, that we make the kind of situation that one oscillation appears and disappears. And so, in such case, only DMD can pick up the difference between the stationary case and non-stationary case. So I think the it is because the DMD calculate including some kind of dimension reduction. So it pick up the some dominant frequency at that moment, and so by using some appropriate dimension reduction, we can characterize or distinguish the two states between the stationary and stationary cases.
Thank you very much. We have one question. How well can a linear model x dash equal ax capture the actual data variance?
That's a great question. Sorry, I haven't systematically evaluate this for these questions, but yeah, we sometimes use the simulation data to how much the simulated data was recovered by the DMD method. And yeah, actually it depends on the data set, but basically we are confirming that we can pick up the frequency kind of very reliably, and also the spatial patterns. So yeah, I think it can work in a practical situation, but I cannot say how accurate it is. Sorry. Is it enough to?
The last question. I understand your method observed a 10 Hz peak in the ADEG, given that alpha waves typically shift to lower frequency in Alzheimer's fiber disease. What is the likely source or nature of this specific 10 Hz activity detected by your technique?
So what's the alpha, the source of the alpha wave? Is it correct? So yeah, actually, yeah. So there's many kind of hypotheses or the data which what kind of brain activity is the source of the alpha, and beside that, and also we found some difference in the more lower frequency range, so the around the theta range, and we don't know the exactly what this means, because we haven't observed this kind of peak in the past spectrum density, so we haven't seen this in the previous analysis. But I think this is corresponding to the some unstable oscillation, unstably appearance of oscillation of this frequency range. So I'm thinking that there's two kind of the changes in the dementia state. One is the appearance of the unstable lower frequency oscillation, and also the instability of the alpha range, the physiological existing oscillation. So there might be some different causes these changes of the oscillation.
Thank you very much.
Thank you very much for inviting me here, and I'm very happy to communicate with AI people. Honestly, I have to confess that I myself is not the AI researchers. and I'm not very good at mathematical analysis or modeling, but I'm just a stupid engineer and just driven by curiosity. So my talk today is about the brain characterized as a physical reservoir, because it seems that many people are very interested in the dynamics of the brain. Yeah, so my original background is the mechanical engineering. When I was a grad student, I made a microelectrode array by myself and bring it to the medical department and ask the doctors to have my electrode implant in the rats or cats. That's how I started neuroscience. And I realized that rather than making these kind of scientific instrument, I'm very happy when I make physiological experience by myself. So that's why I started recording with the multiple electrode array and also making a behavioral experiments in one hand. And also on the other hand, we have some collaborator who is very good at CMOS array development, and we investigate the neural activity at this very small scale. So the scale I have handled is that around 20 micron. So this scale means the inter-electrode intervals, and also 1 centimeters, such as EEG. And I also develop a method by myself and do some system integration to make a biosensor, or just operate a neural robotics, or sometimes diagnostic system. So my lab is doing the research at 3 levels, and in the first level is the in vitro. It's we use a dissociative culture of neurons on the CMOS array, and how the activity develops in the petri dish, and use it for some gameplay. And in the second level, we do our rodent studies in order to understand the biointelligence. We condition the rats and how the brain works, or recently we are interested in social interaction. And the third level is the EEG and fMRI. We are doing these one for to make some brain-machine interface. Yeah, so this is why I'm here. So one of the organizers sent me an email, said that I'm currently in a bit of Chuni disease state. The application of deep learning has been rapidly advancing over the past decade, and data-driven research will almost be replaced by it in the future. I'm wondering if this is really the right way to go, and how to understand the relationship between brain dynamics and brain function, so that I can feel like I understand. So I would be grateful if you could suggest some suggestion at the workshop. This is the request by the organizer. And I asked the generative AI that my friend is suffering from Chunibyo, and the generative AI said that Chunibyo is only a phenomenon peculiar to the adolescent, not a disease. It is important to keep a warm eye on them. So I sent this message to him, and that he said that it was more like a midlife crisis. So it is very, very severe, I think. But maybe this is not a matter of laughter, but maybe many people are suffering from that kind of feeling. So Hinton said that when maybe I think it's 2 years ago, he said in the interviewer asked that what's the worst case scenario that conceivable, and Hinton asked that it's quite conceivable that humanity is just a passing phase in the evolution of intelligence. I think this is a very, insightful words, and probably humanity includes how the brain works. So, yeah, that's how what I felt from these words. So, for example, so yeah, I'd like to talk about why we are interested in biointelligence previously. And yeah, so I asked my lab colleagues to bob their head in synchronization to music. Something like this. Yeah, he's a very good prediction. He has a very good prediction about the musical rhythm. And she's a bit different. She has a bit different prediction system of the music, but still she has a very good predictor of the musical rhythm. Yeah, she's a bit chaotic, but still a good predictor. And we are very interested in music because the music stimulate our prediction system, and that can, induce some rewards. But probably no animals can have some prediction system like this. So we believe that the music is a good research target. But we recently found that something like this. So we present the music to rats, and the rats do exhibit some beat synchronization behavior, or music modulate the rats' behavior. And this is very interesting. But what is more interesting to me is that humans are very good at head bobbing in synchronization to the original music tempo. It's around 120 BPM. But when we increase the tempo by 4 times, it is very fast. And yeah, I myself tried it, but it is very, very difficult to bob their head by prediction of the very fast music. And this was also the case in rats. So therefore, music is not a good proxy of prediction system, but music just demonstrates limitation of our predictive ability in the brain. So, but why we are good at head bobbing at only within a limited range of around 120 BPM? So we inserted the microelectrode array to the rats. It's a 10 by 10 microelectrode array in the auditory cortex of the rats, and presented music and how the neural activities synchronize to the music. And we found again that the original tempo has a very nice beat synchronization, but if we increase the tempo, then it decays. It's very consistent with the behavioral observation. So maybe the brain itself has it's a 120 BPM is tuned. So why is this? So we make the situation more simple. In this simpler situation, we presented a galloping rhythm. It's a click, 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 rest, click, 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 rest, and it's in 60 BPM. It's a very slow, and 120 BPM to 480 BPM. And what we observed here is that if the tempo is very slow, click, 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 rest, something like this, then it's sufficiently slow so each click can elicit the same level of neural activation. But if we increase the tempo, like 480 BPM, it is too fast to follow to the rhythm. And then each rhythm could not activate neural activities sufficiently, and something like this. But in between, the first click has a largest responses, and this response suppress the second one, and the second one suppress the third one. This is why we have a 120 BPM tuning. So adaptation, this kind of adaptation dynamics might underlie the beat synchronization. So we made a mathematical model of this adaptation dynamics. We found that this kind of kernel of the adaptation can well explain the experimental data. So this kernel means that the stimulus within stimulus within the 250 millisecond can suppress the subsequent neural activities. This is very simple, but very powerful to explain the 120 BPM tuning. So this kind of intrinsic dynamics has a constraint of prediction ability. So this kind of result is very consistent with the notion of reservoir computing, which is recently caught attention in the field of AI. And in the usual AI, we optimize the parameter in the network, all of the parameters of the network. And the optimization may be through back propagation, and all of the parameters are subject to change. But in the reservoir computing, we don't care about the parameters within the network. They might change or might not change, but we don't care. We just optimize the readout. And if this network has rich dynamics, then we can reconstruct some function between input and output. This is the reservoir. And in other words, instead of optimizing the whole network, the reservoir computing just takes advantage of existing dynamics of the network. So when the reservoir computing was first proposed, it was surprising that this kind of neural networks, very simple neural networks with rich dynamics, can do better jobs than expected. And this is very compatible with the neuroscience, because the reservoir is not subject to change, and we do not need to optimize there. And any dynamical system like the brain can serve as a reservoir. So such concept is called a physical reservoir. And the idea is that to consider reservoir as a physical reservoir. So the dissociated cultures plated on the petri dish, or the auditory cortex measured by this kind of microelectrode array can be served as a reservoir. And my question is how to quantify the performance of this reservoir. In the classical reservoir studies, it was used some benchmark task, and they discussed that this reservoir can solve this kind of task, but not this kind of task. But it's not a comprehensive measure of the performance of the reservoir. So we use mathematical measure called information processing capacity, or IPC. So think of input sequence of U, which is temporally uncorrelated. And the objective is to construct a target function Z of U. And if we can, if we present this input to the reservoir, then the reservoir have some neural activity with hopefully with rich dynamics, and we can make a linear regression, and then reconstruct this target function. And if this target function can be constructed in terms of mean square error, then we can say that this reservoir can have a IPC of Z. So the definition is something like this. And the idea is that we make the many Z's with orthogonal polynomials so that we can sum up all of these IPCs. And yeah, so we put the input sequence to present some input sequence, and then the brain have some dynamics, and then see how IPC can be obtained with this definition. So we do this in the rat's auditory cortex. We have implanted the U-directed array and presented click sequences. Click sequences is one or click or non-click, so blue or red. And when the click was presented, then we have some evoked responses like this. So with this definition, we found that some IPCs in the auditory cortex. The good news is that the IPC was not zero. And this figure shows the breakdown list of the first and second order IPCs for various interstimulus intervals. So because yeah, so the brain has optimal dynamics. Maybe the optimal dynamics is around 10 to 20 milliseconds. And according to the first order IPC, the input sequence are memorized in the auditory cortex in a linear manner, maybe within about 10 steps. And this panel shows that the second order IPC, indicating that the auditory cortex had non-linear memory with a combination of n and n-1 step memory. So if we have this kind of IPCs, then we can use the auditory cortex in order to solve the logical operation like this. So we attempt a time delay logical operation using the auditory cortex when the input was given as a sequence of 1 and 0. And we use the auditory cortex to derive AND or XOR of the last first and the second step. And of course, many times it's not very accurate, but it is still better than the chance. So like this, we can quantify the brain with the as a reservoir. So it was very surprising that because the rat was housed in a very quiet place, but still they have a information processing capacity. Then how we house the rats under a very acoustically rich environment. So we house the rats under music exposure. Yeah, so Lucky Group has this kind of original piece of music, but for unlucky groups, they have disorganized music. The rhythm structure is preserved, but the pitch was shuffled. And here, the rhythm is shuffled. We presented this kind of music 8 hours per day and house under music exposure for 4 weeks or more. And surprisingly, I'm not, this is surprising or not surprising, but this kind of acoustic environments increase the information processing capacity. So here, N is the naive rats, and this is the breakdown list of the IPC, first, second, third, and more. And this one is the summing up of the total IPC, and N means the naive rats. This one is the exposure to the original music, and this one is the pitch shuffle, disorganized music, and this one is a rhythm shuffle, disorganized music. Like this, irrespective of the music structure, but the sound, probably not the music exposure, can increase the IPC, particularly in the non-linear forms of information processing capacity. So there is a historical myth that music is very good for the brain. And probably the logical thinking is something like this, because the musical rhythm is so beautiful, and that kind of musical structure can be copied to the auditory cortex or the brain, and this is very good for the brain. But this was not the case because the disorganized music have a very similar effects in the development of the IPC. So I'm recently thinking of the cause effect is the reverse. That is, we have an intrinsic structure of the information processing in the our brain, and this is the source of the music. So here's my take-home messages. Yeah, probably not only Yamashita-san, but we are all experience a kind of midlife crisis or identity crisis, because much of the research is we used to find interesting. For example, arts and fine arts and music, or something like consciousness or social interaction, etc. Merely examines how imperfect the brain was, rather than a proxy of intelligence. And this is because agents with perfect intelligence, probably we are seeking, do not need actually the fine art and music and consciousness or social interaction. But however, if we want to live happier, then rather than creating perfect intelligence, I hope that this kind of dynamics of the brain will become a more and more important research topic in the pursuit of humanity and probably happiness. Okay, so thank you for your attention, and this is my team.
Thank you very much for your very interesting talk.
Okay, thank you for the introduction, Lisa. So since I heard that this workshop is about a progress report about GEM, so I'd like to talk about what I did as a research scientist at RIKEN AIP Center. So basically what I did was development of new frameworks for nonlinear replacement learning and its application to brain image data. So let me start from self-introduction for myself briefly. So until the end of last fiscal year, I was working at RIKEN AIP Center as a research scientist. But from this April, I'm now working at Shiga University. And basically, in my talk today, I'd like to talk about what I did as a period of research scientist at RIKEN AIP Center. And during this period, I was working on the development of new analysis method for brain measurement data, especially based on nonlinear representation learning. Okay, so let me start from the background. So especially in recent years, we have better access to openly accessible big neural imaging data. And by using those datasets, we are expected to understand the fundamental mechanism of our brain and then develop practical applications based on that, such as biomarkers and so on. But still, there are huge gaps between them. And I think the difficulty comes from the difficulty of analyzing those datasets. For example, neuroimaging data usually has high dimensionality. And as you know, brain has very complex and nonlinear dynamics. And especially in the case of neuroscientific experiment, the amount of label information is very limited. And there exist many different types of variability, such as variability across subjects, variability across time or days, or measurement size, and so on. So those difficulties are actually making those practical applications very difficult. So in order to fill those gaps, in our project, we are trying to develop a new framework of representation learning, which extract fundamental low-dimensional representation hidden behind data in a data-driven manner. So, and actually, the conventional linear model wouldn't work because of those problems. So especially we focused on nonlinear type of representation learning. So that is what I was doing. So what is representation learning? This problem is basically a kind of problem to extract the fundamental representation hidden behind the data. This is just a schematic image of this problem. But for example, we want to find this kind of low-dimensional space from behind the data. And on this space, in this figure, a single point corresponds to a single data point. And by embedding the dataset in this low-dimensional space, we can somehow understand the hidden structure. For example, around here, we can find samples related to automobiles. And around here, we have samples related to animals, especially with level of horse or dogs, and so on. So by embedding data into this kind of space, we can understand the structure like this. So this is what we want to do. And of course, depending on what kind of dataset we use, the structure we get are somewhat different. For example, if we input MNIST into VAE, we obtain this kind of two-dimensional structure in the latent space. And for example, we can see such as the similarity between digits of this space. That is also a structure of the dataset. And if we input facial images of celebrities, we, for example, obtain some this kind of meaningful information. For example, this component represents whether the images were wearing sunglasses or not. This axis represents difference of hair colors also. So there are many practical applications of this representation learning. But unfortunately, representation learning is not always an easy task. So this is the mathematical formation of the representation learning. We have observation X, and we want to estimate latent variables from the observations. And this latent variable is generating the observation as unknown observation mixing like this. And the ideal property of this model is called identifiability. In other words, uniqueness of the solution. If we can identify the parameters from the data distribution uniquely, we say that the model is identifiable. This property is crucial for justification and interpretation of the model. And also, this property is known to be important to achieve good generalization performance. However, since this problem is fundamentally an inverse problem, we cannot say this property without any assumptions. And for example, in the case of BAE, we assume IID samples are the mutually independent Gaussian latent component. But those assumptions are not enough to guarantee this property. And this is just a visual illustration of this problem. So this latent component satisfies this assumption of BAE, IID mutually independent Gaussian latent variable on two-dimensional space. And let's say we observe observations as linear mixing of these latent components. Then let's say this is the true model. Then next, let's consider a false model, where we simply applied rotation to the two latent components. But in this case, this rotation doesn't really change the distribution of the latent components, which means it doesn't affect the distribution of observation either. So we cannot really say which model is correct only from this observational distribution. In such case, we say that the model is not identifiable. And it is well known that in the case of linear model, we need to assume non-Gaussianity of the latent components. But in the case of nonlinear model, the problem is more complicated. The assumption of the distribution of latent component doesn't work at all. But fortunately, we found a solution. By additionally assuming some kind of data structure, additionally, we can guarantee the identifiability. More specifically, instead of the unconditional mutual independence like this, we assume conditional mutual independence given additional variable u. And we also assume that this u modulates the distribution of each component significantly. And depending on what kind of u we consider, we can consider different type of temporal structures like this, such as non-sessionality or autoregressive signal, or more general case, and so on. The important point is that those assumptions make the model identifiable, then we can estimate the latent variables properly, like this, even when BAE doesn't work at all. And the other interesting point is that by using those data structure, we can propose new type of estimation framework based on self-supervised learning. For example, in the case of non-stationary time series, we train a nonlinear feature extractor and a classifier to predict segment level. Then after such training, the output of this feature extractor represents the latent components to some extent. And in the case of autoregressive time series, we train nonlinear feature extractor and a discriminator to discriminate real data and temporary shuffled data. Then after such training, again, the output of this feature extractor represents hidden latent components to some extent. The idea is very simple, but the point is that from the original unsupervised problem, we can convert them into supervised problem by utilizing the data structure assumption. So this is the important point. And we also applied those frameworks to brain imaging dataset, for example, image dataset. And here, especially, we used the non-stationary type of nonlinear RCA. And we found, for example, hierarchical spatial coactivation patterns from reference data. And also, we found that the generalization performance of the extracted components by our framework has better generalization performance compared to the basic baselines. Okay, so far, so this is kind of background because those projects were conducted before I joined the RIKEN AIP. Then from now, I introduce what I did during research scientist as RIKEN AIP. The first project was an extension of those temporal ICA to spatial ICA. The idea is actually very simple. So this was just a flipping of time and space axis. But this structure was it's kind of reasonable, especially in the case of neuroimaging data, because the amount of temporal step was much limited compared to spatial dimension. And also, assuming spatial non-stationarity is also reasonable, especially in the case of brain activity. And by applying this framework to big brain image dataset, like Human Connectome Project dataset, we found what we called temporal primitives of ephemeral signals, which means those primitive patterns were actually generating the ephemeral signals as convolutional way. And we also found kind of nonlinearity of those temporal primitives, and also the spatial co-activation patterns of fMRI were basically organized by those temporal primitives. These were interesting findings. And the other project was an extension of nonlinear shade to dynamics. So in usual setting of representation learning, we assume instantaneous mixture, which means we observe observation from components for each data point T. But we found that actually we can generalize this model into dynamics. Here we have the information in the past inside of this function f. So in such case, we can interpret this f as dynamics, and this latent component s can be interpreted as a kind of external factor affecting or controlling the dynamics. And what we found is that from this model, we can estimate this s from time series x without knowing this dynamics f by using the similar idea to nonlinear share. The assumption is very similar to nonlinear share. We just need to assume conditional independence and non-stationarity of this latent factor S. And we can also propose a new estimation framework based on self-supervised learning, similar to nonlinear share. And we apply this framework to real image dataset during auditory and visual stimulus. And we applied our framework to 148 channel image data obtained from 20 subjects during such experiment. And these are the 5 components extracted by our framework. For example, this component 1 seems to be related to auditory stimulus from this temporal and spatial patterns. And this component 2 seems to be related to visual stimulus from this temporal and spatial patterns. And those components seems very reasonable in the aspect of the neuroscientific knowledge. And we also found that the subject transfer decoding performance was much higher compared to the baselines. So this indicates that our framework could extract representation, which is very similar across subjects. And we also worked on the project of causal representation learning, or CRL. What we want to do in this CRL is like this. Given a set of observations, we want to extract this kind of representation. For example, in this dataset, we can see that there are 9 animals, 9 dogs here, and they are moving based on reader flow network like this. So this problem is a kind of combination of 2 problems. One is, of course, representation learning. finding of these abstract nodes, in this case animals, behind the data. And the other one is the causal discovery, finding of the causal relationships between those abstract nodes, the blue arrows in this figure. So in the case of CRL, we need to perform both of them simultaneously. But since both of those two problems are ill-posed and difficult to solve, this CRL problem should be much more difficult. And the difficulty comes from this assumption of the latent variables. The observation model is the same with the general replacement running, but the latent structure assumption is different. In usual replacement running, we assume mutual independence of latent components, but here we assume kind of dependency between variables. So of course, this is better in the sense of the representation power of the model. The problem is that this assumption is much weaker compared to the independence. So because of this, we cannot guarantee the identifiability for the model, and also we cannot estimate properly. And then we found a solution to that. So we found that by assuming a specific type of observation model, we can guarantee the identifiability. More specifically, we assume that we can divide this observation model into multiple groups like this. In this figure, we have 3 groups. And observations are obtained separately for each group, group 1 and 2 and 3. Then we additionally assume some causal relationship assumption based on pairwise relationship like this. Then based on those assumptions, we found that we can identify the model and estimate the model. So there are many possible practical situations where we can use this model. For example, in the case of neuroscientific field, then let's consider the case where we have much model data of brain activity and external stimulus and behavior data. We can estimate the causal relationship between those modalities in latent space. And in the biomedical data of the combination of life log and genetic factor and disease diagnosis data, we can possibly find a causal relationship between those factors. There are many possible applications. And also we proposed practical estimation framework consistent with the model. And from original dataset, we prepared artificial dataset where intergroup structures were broken by shuffling. Then we trained a logic regression classifier to discriminate those two datasets. Then after such training, we can say that the feature structure represent the latent factors, and this weight parameters W represent the causal graph, HIC matrix lambda. So this estimation framework is very similar to the idea of nonlinear shape. And we applied this framework to high-dimensional simulation data. Here we considered that we have 2 groups, and for each sample, we obtain a set of images of a port like this. And the point is that those images are generated based on 10 latent factors, such as 3D location and 3D rotation of the port, and difference of color of port, background, light, and location of the light. And those factors were causal related between groups and also within groups. And the goal of the simulation is to estimate those latent factor and causal relationship only from those observations. And the problem setting was very difficult, but still by using our framework, we could estimate latent factors and also latent causal structure like this. So there are many possible applications for this framework, but so far I haven't tried application. Well, I tried some applications, but I still haven't finished. So, and also I'm looking for interesting application of this framework. So if you know good target data, please let us know. Okay, so let me summarize. The research goal of our, this team, the computational brain dynamics team, is to develop biomarker, next generation biomarker based on big new image data. But there was still a huge gap between them. So my project was basically developing new framework of representation learning. Especially we are working on the identifiable type of representation learning based on some structure assumptions. And we also proposed a special framework, consistent model. That is what I was doing at RIKEN AIP. Yeah, that's all my talk. Thanks so much.
Thank you very much, Morioka-san. So for all the audience, if you have any questions, please ask in the Q&A box or raise your hand. We have about 2 or 3 minutes. So Morioka-san can ask a question. So in your framework, the representation is a very important concept. So for example, for the brain data, because the representation is very abstract, so what kind of way do you have to examine whether a representation is good or not?
Well, yeah, that is very good question and a very important question. And well, you know, although the frameworks I proposed has theoretical guarantee of identifiability, still the problem is that in the case of, especially in the case of nonlinear representation learning, it's very difficult to interpret what kind of components were extracted from the data. So it's kind of difficult to evaluate how those components are good in the sense of the neuroscientific knowledge. So, well, of course, you know, one possibility is to apply, I would say, use the extracted components to some kind of downstream task, like subject transfer, decoding, or something. Then, if the performance is better than the baseline, possibly we can say that the representation somehow capture proper brain dynamics compared to baselines. But still, it's kind of difficult to directly evaluate what kind of components were found by, in the case of nonlinear learning framework, and it's difficult to justify practically. But that is very important question, and I need to work on that direction at some point. Yes.
Okay, thank you very much for your answer. So if anyone has some other questions, please input your question in the Q&A box, and Morioka-san, please answer all the questions in the Q&A box. Okay, it's time to welcome the next.
All right, thank you so much for having me at this workshop, and thank you all in the audience for coming. My name is Daniel Muller-Komorowska, and I am a postdoctoral scholar at the Okinawa Institute of Science and Technology. And today I will be presenting you my work where I use simulation-based inference to understand the mechanisms that can cause and hopefully prevent unhealthy types of dynamics that cause epilepsies. So to begin with, I want to give a brief introduction on epilepsy. Epilepsy patients suffer from recurring seizures that cause loss of consciousness, convulsions, and various other very debilitating symptoms. A seizure itself occurs very suddenly within milliseconds or seconds. But there is a slower process going on that is called epileptogenicity that occurs over days or months. when brain areas change their excitability in a way that makes seizures more likely. And that's kind of the part I'm most interested in, these kind of slow changes and excitability more generally. And I use spiking microcircuit simulators to study the neuronal network mechanisms that maintain or disrupt healthy types of excitability. But before I talk about the details of that, I want to briefly explain something that I think is very important for not just epilepsy, but brain disorders in general, and that is non-uniqueness, which is a fundamental kind of property of brain disorders and possibly brain function. And I want to illustrate it with this very simple cartoon where, let's say, we just have two parameters because those we can visualize very easily. And let's say in this orange region here in the middle, that's where we want to be if we want to be healthy. And there we have a very low chance of suddenly having a seizure. And an epileptic patient, they've transitioned from this orange region into this black region. And then they present to their neurologist with problems. So let's say we have two so we have two patients, and they're both they ended up in the same black region. They're both they probably also present to their doctor with very similar issues, but they've started in completely different places and they've took a completely different direction to where they are right now. And if we think then of anti-epileptic medication as a perturbation that brings people back into these healthy regions, it's very clear that we really would ideally want to know not just where the patient ended up, but also how they got there and what are the what is the entire non-unique space of being healthy and unhealthy. And in real patients, that's of course extremely difficult because there's hundreds, thousands of parameters, many of them probably still completely unknown to us. And even in a computational model, you might imagine that this is quantifying this non-unique space is extremely difficult. But I hope that during the talk today, I can convince you that we now have techniques that we call simulation-based inference that allow us to quantify this kind of space for neuronal network models that have about 30 free parameters. So, as I said, this technique I try to use to quantify this kind of degenerate space. Degenerate is a different word for non-uniqueness. So this non-unique space is called simulation-based inference. And the idea is that we have a simulator that's simply a computer program that takes some kind of parameters, theta, as input and generates some kind of output from it. And simulation-based inference wants to solve the inverse problem of that. And that's instead of taking parameters and generating data, we want to know, given that we already have some kind of data, or we already we know how the activity in our epileptic patient looks, or we know the average epileptic activity and the average healthy activity, what are the likely parameters? What are the parameter distributions given that data? And that's, of course, related to Bayes' rule, where this parameters given data is essentially the posterior. And if we knew the likelihood, this problem would be relatively easy to solve. But the problem is, if a simulator is stochastic and complex, we usually don't know the likelihood. But it turns out that, and that's a relatively recent advance, it turns out that there are ways to estimate the posterior distribution without relying on the likelihood. And one of those ways is neural posterior estimation. So neural posterior estimation is based on relies on a simulator because it's a simulation-based inference technique, and we need to define our simulator and the prior distribution of the parameters. And then we sample randomly from the prior, ideally millions of times in a way that slightly oversamples the prior distribution. And then that generates output. For each prior parameter pair, we get output. And then we can use this as training data for a neural density estimator to train it to directly estimate the posterior distribution. And that's kind of the major advance in this simulation-based inference field, was to show by other people, not me, but other people have shown that there are learning rules for which if you do that you actually they've proven that you target the true posterior of the simulator. And then once the neural density estimator is trained, we can take observations, plug them in for our X, and then we get the posterior distribution of the parameters given that observation. And if we take parameters from the high likelihood region of that posterior, we simulate stuff that looks very similar to our the observation we conditioned on. And if we're outside, then we get something completely different. And if we don't have confidence that this posterior estimate is good for some reason, this posterior estimate can be plugged in as a new prior, and then we kind of sample more efficiently and can repeat that step, and that's called sequential neural posterior estimation. Now, I want to briefly, though, talk about, because we need to, for this, we need to simulate millions of times. So we cannot really use very biophysically realistic simulators. But I still wanted to have some kind of biological realism of kind of phenomenological spiking neurons. So what I'm using is adaptive exponential integrate and fire neurons with Todux-Markram synapses. And the simulator specifically has one excitatory population, two inhibitory populations that have different, these neuronal populations have their own kind of free intrinsic parameters that can be subject to simulation-based inference. And this on the left here is just an example of how the adaptive exponential integrate and fire neuron processes inputs. So what I've done with this technique is I've just first tried to create kind of what I imagine as a baseline activity level and kind of a hyper-excitable activity level. And to do that, I've defined 7 summary statistics: the mean entropy, the mean rate, the gamma power, theta power, fast power, the interspike interval, the CV of the interspike interval, and the correlation of the spiking. And then I just targeted something that has relatively low activity. That's kind of the what I imagine as the healthy state. I call it baseline. And then something that's clearly hyper-excitable that is very unhealthy because neurons are silent for most of the time, and then suddenly they become extremely active. And these are simulations that show that we can generate simulations that that target these points, these kind of feature points. And then we can create these posterior distributions given these kind of conditions. So this green one indicates that we have a posterior distribution that should generate simulations that have these kind of properties in green, and then the hyperexcitable one should generate simulations that have these properties in orange. And to verify that this worked, I use something called posterior predictive check, where in the X marks the point that we're targeting, and the box plots show actual simulations where the parameters are sampled from the posterior distribution. And this just shows that we're relatively good on target. There's some slight miscalibrations here in the gamma power, but for the most part, at least the baseline is clearly much less excitable and more kind of dynamic than the hyperexcitable one. And what can we do with this posterior distribution? We can, for example, look at the pairwise correlations. So here we have the principal cell voltage threshold and the principal cell equilibrium conductance of the equilibrium potential of the leak conductance. And we can use these pairwise correlations to discover compensatory mechanisms that maintain this kind of dynamic, whatever we plugged in for the X, in this case, the baseline. And what we see here is that if we were to increase the voltage threshold, we leave this X-type dynamic. But if we want to restore it, we can increase the equilibrium potential in turn. And that's how the posterior distribution reveals compensatory mechanisms. And that's just one example. The actual simulator has 32 parameters. And this is how it looks, where the marginal ones are on the diagonal, and then all the pairwise correlations are in the upper triangle. And I will not go into all the details of these, but there's one more thing we can do, and that is, instead of just having the X, we can introduce an additional condition. So we can set another parameter to a specific value M, and then we can set that parameter to a different value, and that allows us to specifically investigate the compensatory mechanisms in response to different perturbations. So for example, if you plug in different interneuron numbers for this parameter, we can have a normal number of interneurons and a lower number of interneurons. And if we then look at the posterior distributions, theta, of the other parameters, for example, here is the connection probability from adapting interneurons to principal cells, we see that to maintain both want to maintain the X-type activity, the X-type dynamic. To achieve that, in the when there's interneuron loss, this parameter increases. And we can do that with all the parameters and the different conditions where we have interneuron loss, excitatory recurrent sprouting, and intrinsic hyperexcitability. And we see that for these different conditions, there are different they differ in which parameters have very good compensatory potential. For example, for interneuron loss, it's the connection probability from adapting interneurons to principal cells. Interestingly, the non-adapting interneurons have less compensatory potential in that regard. For intrinsic hyperexcitability, it's the voltage threshold. And for sprouting, that's somewhat trivial because sprouting is an increase in the probability of recurrent excitatory connections that can be compensated for by decreasing the strength. So but there's also, interestingly, the second strongest is an intrinsic property, the leak conductance. So that's kind of where I'm at with this project. I want to briefly say what am I currently doing to expand on this. Well, you might have noticed so far I only have two conditions. I have the baseline condition and the hyperexcitable one. And that's because I wasn't able to build kind of an amortized estimator. So what I needed to do is for the baseline, for the hyperexcitable one, I needed to do this loop, I think 4 times to arrive at a good posterior estimate. So now I'm running more simulations. I'm changing the architecture of the neural density estimator to arrive at an amortized estimator, and that will allow us to not just look at the compensation for this one baseline condition, but for the kind of sensitivity of parameters more broadly across the whole kind of all possible observations, basically. So that's what I'm currently doing. And there was some interesting talk today, a lot of mention of biomarkers. And I'm very interested in biomarkers of diseases, of course. And I want to highlight that if one of these parameters is kind of in interest, is somewhat related to an interesting biomarker, for example, the number of interneurons and the inference. So my current work is theoretical. I'm now working on integrating patient data for another project, but what I've presented is not based on patient data, but I'm now working on integrating electrocorticogram data. And if the data comes from a specific patient, the posterior kind of reveals a biomarker. And it could even reveal compensatory potential for that biomarker. So it reveals, with respect to the simulator, kind of a mechanistic biomarker, an actual mechanism that could change the activity. And with that, I want to summarize. And I want to the take-home message for my talk is that simulation-based inference estimates the non-unique parameter space that produces healthy unperturbed neural network dynamics. Posterior parameter correlations reveal compensatory mechanisms, and the efficacy of different compensatory mechanisms depends on the underlying perturbation. And with that, I want to thank my supervisor, Tomoki Fukai, and his neural coding and brain computing unit, and the scientific computing and data analysis core facility at OIST that provide the high-performance computing that I rely on, because, as I said, I need to simulate millions of times, and so I need a lot of CPU cores. And this is kind of the preprint that describes the results that I've shown you today. With that, thank you for your attention.
Okay, thank you very much, Muller-san. So we welcome any questions from all audience. Of course, we still have 5 minutes. So first, I want to have a question from the viewpoint of application. So at the beginning of your presentation, you mentioned, so different patients, even with the same disorder, they may have different reasons. So after you estimate the parameters of the neuronal network for the patient, so what kind of method should we use to design like personalized treatment for different patients?
What kind of method? So we don't know yet, right? It's very hard to say. That's a really big question. But the way I approach it is that I approach it from the simulator perspective. So what kind of parameters does the simulator expose? Because there will always be biomarkers that you can kind of just get from the actual data, right? You look at the power spectral density, or you look at the some kind of decomposition, and you say, oh, this is this patient at this kind of frequency and things like that. But now we're looking at these kind of simulators, and one thing I'm, for example, interested in is interneuron number. So in some epilepsy patients, they're sclerotic, and some epilepsy patients are non-sclerotic, and you can kind of see that in the MRI. But I would be interested in, are there ways that we can determine what's the number of interneurons that are actually in that patient's network, like at a specific electrode? And that could kind of pave the way for things like more targeted anti-epileptic drugs that target something. But
Okay, we
yeah.
still have several questions. So first, it's a quick question. How many runs on average did it take to get a good posterior distribution?
What was the question? In the chat, I didn't hear.
Yes, in the chat.
How many runs average did it take to get a good posterior? I did not get an amortized posterior with about 1.5 million simulations. But it really depends on, like, that's impossible to answer. I can give you the answer for my simulator, but it really depends on the number of summary features. So I just have 7 summary features, which is very little, but I have 32 parameters, which is a lot. Like, people usually say you should not use simulation-based inference with more than 30 parameters. So I was in the millions, and I did not get a good amortized posterior. I'm still running simulations. I will go into 10 millions and hopefully get an amortized one. And then for the sequential one, I think I did 4 loops and each had 20,000 simulations. So the sequential one is more simulation efficient because you're not using this kind of broad prior, but you are using the more targeted posterior to kind of sequentially zoom into the true one.
Okay, your talk is really interesting because there are so many questions in the
I can
Q&A
answer
box.
them in chat.
Yes.
Thank you very much.
Hi everyone, I'm Zijian Dong, a final year PhD student from National University of Singapore. So thank you for having me here. I would like to present our work titled Brain-JEPA, Brain Dynamics Foundation Model with Gradient Positioning and Spatial Temporal Masking. So nowadays, AI is experiencing a paradigm shift from task-specific training to building foundation models that are trained on extensive data using self-supervision at scale. So unlike the models with singular functions, so foundation models can be adapted to a diverse array of downstream tasks. So here we are interested in brain fMRI signal, which is a type of non-invasive recording with high spatial resolution. So many questions remain along the journey towards brain foundation models using fMRI. So in this work, we specifically address 3 critical questions or challenges. So in a transformer-based system, so how can we locate different brain RIs in the model? During the pre-training, does direct reconstruction of mask fMRI input make sense? And how to efficiently mask fMRI input during the pre-training? So now let's see the first question. To address the challenge of locating RIs in transformer, we introduce brain gradient positioning, using brain gradient as a functional coordinate system for brain in attention-based models. So we begin with fMRI time series data, which shows us how neural activity fluctuates over time in different brain RIs. So from these time series data, we can construct a functional connectivity by calculating correlations between every pair of regions. So in this functional connectivity matrix, each row or column represents the connectivity profile of that region. We can then calculate the cosine similarity between different connectivity profiles, leading to an affinity matrix that quantifies how similar the overall connectivity profiles are between different brain regions. Afterwards, the functional connectivity gradient is then calculated through the diffusion map of this affinity matrix, which is a continuous measure that captures the functional relations among different RIs, where relative distance between different RIs indicates the similarity in their connectivity. So now let's look at this figure. So we can see that the brain cortical regions are situated in the top 3 gradient axes and colored based on their positions. So if you look at the red panel, each dot represents one brain regions. And if you map the colors back to the brain surface, we can see the functional network organizations such as default mode and visual networks. So the second question is that during the pre-training, does the reconstruction of mask fMRI input make sense? So here let's see 3 common self-supervised learning architectures. So the first one is called drawn embedding architecture. So imagine you want to train an image encoder given an image input, for example, the X here. You can apply different deaugmentations to it to become Y. So for example, you can perform scaling, rotation, or cropping of this image. So the assumption here is that given one source, so the one image here, differently augmented images should share some invariant semantic features. So for example, given a photo of a dog, no matter how you rotate it or scale it, it still be a dog. So the training here is really to maximize the similarity between embeddings from different views of an image. But this architecture faces a fundamental limitation when applied to fMRI time series data because it relies on handcrafted augmentations that may work well for images but not suitable for fMRI time series, since in this field we still don't have a very well-established deaugmentation strategy for fMRI signal. So the second one is generative architecture. So one of the most famous examples you may know should be the masking autoencoder, in short MAE. So here the input X can also be an image. Before input X to the model, you can randomly mask out some patches within this image, and the training goal is to reconstruct the whole image through an encoder-decoder architecture. So if you apply this architecture to time series data, during the pre-training, it attempts to reconstruct every bit of fMRI time series, forcing the model to capture both the meaningful neural signals and the noise, which is suboptimal. So in this paper, we adopt the drawn embedding predictive architecture, in short JEPA, where it predicts the representations of various target blocks rather than reconstruction of the mask input. It can enhance the higher level abstraction and boost the scalability and efficiency. So for example, here the X can be the input, which is the part of the input data, which is the observation, while the Y is some other part of the input data. So the training here is to predict the representation of the Y given the representation of the X. So the next question is, during the pre-training, how should we select the observation and the target? So as fMRI data has complex patterns across both space and time, we designed spatial temporal masking to inject a strong inductive bias during the pre-training. So after patchifying the fMRI time series data into different patches, we can mask the data into 3 different regions, including cross-RI, cross-time, and double-cross, forcing the model to predict across different brain regions and time points. So for example, you can random sample an observation block from the time series data and train the model to predict the other parts of the time series. So this structured approach can lead to faster convergence based on our experiment and better downstream performance compared to random masking. So now let's see the overall architecture of Brain-JEPA. So it predicts, during the pre-training, it predicts brain activity representations in latent space. Specifically, given the observation block, the model is trained to predict various target blocks. So we use the brain gradient to position the different RI locations and predict the target blocks given the observation blocks. The model learns through MSE loss, which is calculated between the prediction and the ground truth of the target representations, while the target encoder is updated via exponential moving average of the observation encoder's weights. So now let's see the experimental results. Brain-JEPA was pre-trained on 80% of UK Biobank's fMRI data. On internal tasks using UK Biobank's 20% held out data, our model significantly outperforms previous approaches, achieving better performance in both age and sex prediction. So the baselines compared here span across convolutional neural network, graph neural network, transformer, and the previous brain foundation model, Brain-LM. External evaluation across different datasets, Brain-JEPA shows strong generalization capabilities. On HCP aging dataset, we improve significantly in both demographic and trait predictions. So here the neurotype score measures emotional instability and sensitivity to stress, while the flanker score assesses a person's ability to focus attention and ignore distractions. For clinical applications on ADNI and MECC datasets, Brain-JEPA maintains strong performance in disease diagnosis. The mild cognitive impairment, which is the MCI here, is cognitive decline beyond normal aging, but without significant functional impairment. The amyloid status indicates the presence or absence of amyloid beta plaques in the brain, which is a key Alzheimer's disease biomarker. Importantly, Brain-JEPA was pre-trained solely on Caucasian data. It generalizes well to Asian cohorts, which is demonstrated on MECC dataset. It is an in-house dataset we collected from a memory clinic here in Singapore. So next, let me walk you through some findings across for further analysis. So firstly, we conducted model scaling. So here, if you look at this figure, the y-axis shows the model performance, which should be Pearson correlation for regression or accuracy for classification. So x-axis shows the model sizes. So here we show the number of trainable parameters within one model. So here we use 3 versions of the backbones based on Bayesian transformer small, base, and large, corresponding to 3 different model sizes. So we can see that there is a consistent improvement as model size increases. Secondly, we compare our model with the previous brain foundation model, Brain-LM, regarding linear probing performance. So given a pre-trained model, we can perform fine-tuning to adapt it to different downstream tasks. So here, fine-tuning involves updating all or most of the pre-trained model's parameters by continuing training on a specific task, allowing the model to adapt its learned representations to the data. Well, linear probing, in contrast, it freezes the pre-trained model's weights and only trains a new linear layer on top of the extracted features, which is much more computationally cheaper and helps evaluate the quality of the pre-trained representations. So here, the blue line represents the Brain-JEPA, with the pink line is corresponding to Brain-LM. So we can see that Brain-JEPA demonstrates smaller performance drops between fine-tuning and linear probing. So thirdly, we performed ablation study to compare 3 different positional embedding approaches, including sine and cosine functions, anatomical locations, and our proposed brain gradient positioning. So we can see that brain gradient positioning outperforms the other 2 traditional positional embeddings. So in the last ablation study, we compare our spatial temporal masking strategy with the vanilla multi-block masking regarding the performance over different training epochs. So here, the orange lines represent our spatial temporal masking strategy, while the dashed line show the vanilla multi-block sampling trained for 300 epochs. So here, this figure shows that spatial temporal masking demonstrates faster convergence and better performance compared to multi-block sampling. So Brain-JEPA can also provide interpretation for certain task it performed. So here we take the MCI diagnosis as an example. So we can extract the network level attention patterns that can highlight critical brain networks. And we show that the patterns we found within the model is consistent across both Caucasian and Asian cohorts. And the salient networks we found are aligned with the known neuroscience findings. So to conclude, Brain-JEPA introduces 3 key contributions, including prediction in latent space rather than reconstruction, brain gradient positioning for RI encoding, and efficient spatial temporal masking. These contributions lead to significant impacts, including state-of-the-art performance across multiple tasks, strong cross-ethnic generalization, robust scaling properties, and improved efficiency with better linear probing. So what's next for the brain foundation model for fMRI? In the next phase, we aim to build a multimodal brain foundation model by integrating genetics, text, and other data types that is relevant to brain. And given the pre-trained models, fine-tuning can actually be very computationally expensive, which may not be suitable for certain low-resource resume, like some few-shot setting when you have very limited data, or you have very limited like computing resource, like you have a low-end GPU, you want to employ it in some real-life situation. Sometimes fine-tuning is not suitable. So the efficient adaptation of the foundation models could be a promising direction for future research. So we have proposed a prompt tuning-based method, which you can go and check. So basically, when we adapt the model, we keep the model frozen, but we just add some learnable prompt to adapt the model to different downstream tasks. And in the future, we want to also investigate how the brain evolves over time, capturing key patterns in aging and development. So basically, currently, the current version of our brain foundation model cannot understand like the longitudinal data, given multiple time points of the visit from different participants. The model cannot understand that. So maybe in the future, we can also inject the longitudinal information into the model training so that it can capture the longitudinal changing of different patients or participants. Okay, so that's all for my talk. I'd like to acknowledge the co-first author, Dr. Ruilin Li from our lab, for the great contribution to Brain-JEPA, and my supervisor, Associate Professor Juan Helen Zhou, for her guidance throughout this work. Our research utilized datasets from UK Biobank, ETNI, HCP, and MACC. All experiments were performed using resources from NSCC. Finally, I'm grateful to all our lab members for their support. And we welcome you to explore our lab website and follow us on X to learn more about our work. Thank you for your attention. Happy to take any questions.
Thank you very much for your presentation, Zijian, because you know foundation model is really, really hot in recent years. So we want to invite you to give a talk about your recent work. So for all the audience, if you have any questions, please raise your hand or put in the Q&A box. So first, I want to confirm with some technical details.
Sure.
Please turn back to the question 2 you raised in the beginning of your presentation.
Okay.
So I remember you also use, encoder for maybe fMRI time series, right? In the joint embedding predictive architecture.
Yes.
So I'm curious about, so what kind of result did you obtain by encoding the fMRI time series?
What do we mean? What kind of representation?
So it's also a time series with lower dimension, or it's some quantized label or something
Oh, okay. So you mean like so we input the time series into the encoder. What is the outcome from this encoder?
Yes.
Okay. So basically, the key thing here is the compression. So basically, we want to compress the high dimensional, like this kind of spatial temporal data into a low dimensional representations that can facilitate downstream task. So basically, this encoder is trained to compress this time series into some one-dimensional vectors. So these one-dimensional vectors will be further input to like some linear classifier or regressor for the downstream task.
And also, maybe I missed some details. So here, what's the meaning for Z?
So Z is the condition. So basically, if you want to train such kind of architecture, you want to predict the representation like SY based on the SX, right? But you need to condition on something. So here our condition is the position of the RI. So basically the input X is just part of the fMRI data. So it's like sum of the RIs with sum of the temporal segments. So it's a part of a certain segment of this time series. And the Y is some other parts of the time series. So how can the model know that what is the input and what is the output? So you need to provide the positional information of the ROI for the input and output. So the Z is the condition here. You let the model know that what is the input, what ROIs is contained in the input, and what ROI you want to predict for the output.
So you mean Z is just like some positional embedding?
Yes.
Okay. And for the external evaluation of your method, so what kind of way did you use to adapt to your pre-trained model?
For the main results, like
Yes, for main results.
For the main results is based on the fine-tuning. So basically, given the pre-trained model, we fine-tune this pre-trained model on a certain data set, like HCP aging or ANI.
So I guess the first three models are trained from scratch, and the last two models are fine-tuned.
Yes.
Okay, thank you. So there seems no questions in Q&A box. So Zijian, please also pay attention to the Q&A box. Yes, there is one more question. I want to know computational resource required for foundation modeling. So how many GPU and how long time did you use for training?
Yeah, that's a very important question. So this kind of training is usually very expensive. So actually, we have some follow-up versions of this foundation model, and more can achieve more advanced functions, may require even more resource. For Brain-JEPA, for exactly this one, the model was pre-trained on 4 A100 GPUs with 40 GB memory for the pre-training. So that's the computing budget for this model.
And maybe how many hours or how many days did you use to pre-train the largest model for your method?
I think this is fast, but so basically, our model, the pre-training of our model is actually much faster than some standard transformer-based like an MAE model. So basically, there are two reasons. So the first one is, you see that we don't reconstruct the raw input data, right? So in the JEPA architecture, unlike the generative architecture. So this is more efficient. And also, if you look at the details of our implementation, we employ a technique called a flash attention instead of the standard attention mechanism in transformer, which is much more efficient. So basically, the pre-training of this version of the model only take like less than 2 days, of to train the model on the 80% of the UK Biobank fMRI data.
I think this time is much smaller than we imagined.
Yeah. So if you want to train a JEPA-like architecture and you have like A100 level GPUs, basically it's very efficient. It's not that slow.
Yes, because A100 is not so new in this year because we have more powerful GPUs. So anyway, thank you very much for your answer. Thank you very much, Zijian.
See you. Bye.
Thank you, Morioka-san, for the introduction. In this talk, I'm going to review the journey of our studies on spontaneous brain activity conducted in these 10 years. Spontaneous brain activity is the neural activity that occurs in the absence of specific external stimuli or tasks such as in resting state and sleep. Although spontaneous brain activity seems to do nothing, it is very important. Ideas arose in dreams brought about scientific discoveries such as benzene and the periodic table of the elements. Not only these, spontaneous brain activity is considered a source of human creativity that have generated art, technology, and science. So what does occur in spontaneous brain activity? To answer this question, let me start from brain activity during a task. Suppose that this rectangle represents brain activity, horizontal axis represents time, and vertical axis represents space. When we present stimuli such as phase stimuli, stereotypical spatiotemporal patterns appear after stimulus onset, which are usually called stimulus-evoked activity. Such patterns are formed by propagating activities through anatomical connectivity. When an activity propagated from blue to red region through yellow region and return to the blue region, this propagating activity forms a spatiotemporal pattern like this. How about resting state? In the case of resting state, we don't present stimuli. However, it is possible that spatiotemporal patterns appear after the onset of internal event, such as remember the past, imagine the future, and create an idea. Indeed, recurring spatiotemporal patterns in spontaneous brain activities have been widely observed in various species and regions, such as mouse visual cortex, rat visual cortex, mouse whole brain, and human temporal cortex. Therefore, revealing spatiotemporal patterns in spontaneous brain activities may unveil our internal brain activities, such as consciousness. However, there is a big problem. That is, onset of the patterns are unknown. In the case of resting state, we don't have stimulus or response onset, so we don't know when the pattern appears. About 10 years ago, we developed a method to solve this problem called spatiotemporal pattern estimation, STEP in short. STEP estimate recurring spatiotemporal patterns from resting state brain activity data. It's assumed that resting state data contains several unknown spatiotemporal patterns at unknown onset. In this figure, resting state data includes 3 kinds of patterns, and each pattern appears for several times. But we don't know their shapes and onset. This assumption is mathematically formulated as follows. Resting state data Y consists of the convolution of pattern P and onset time series U. Note that the onset time series U takes binary value of 1 or 0. STEP estimate patterns P and their onset U only from resting state data Y. First, we initialize onset U by random numbers. Once onset U is obtained, we can estimate spatiotemporal pattern P by the least squares method. Using the estimated pattern P, we update on onset U so that the residual error becomes smaller. Then we return to the estimation of P. By repeating the estimation of P and U, we can estimate P and U simultaneously. Furthermore, we extended step to big step. Big step can estimate the pattern with less computational cost than step. Then we applied big step to less distinguished MEG and EG data. Using MEG and EG, we can measure brain activities non-invasively with an excellent temporal resolution on the millisecond order. The MEG and EG were simultaneously recorded. By the way, recently a new MEG device called optically pumped magnetometer has been developed. Because OPM sensors work in room temperature, we can flexibly put them around the head. We developed a method to design the OPM sensor array for accurately estimating brain activity as regions of interest. Anyway, but in this case, we used a conventional MEG system with a fixed sensor array. This is a pipeline of offline analysis. From the MEG and EG data, we estimated cortical current. Then we estimated spatiotemporal patterns from the cortical current using big step. This is a sample spatiotemporal patterns. Within 0.1 second, the activity globally and consecutively changed with time. This pattern repeatedly appears in distinct brain activity. This figure shows power distribution of the patterns averaged across the subject. The pattern had large activities around the cingulate cortex. These areas. The activity of these areas, cingulate cortex, were reported to reduce during unconsciousness. So the pattern may reflect consciousness. To elucidate the generating mechanism of the patterns, we conducted a simulation test. We constructed a whole brain dynamics model using Kuramoto oscillators and simulated spontaneous brain activities. This figure shows the similarity of the patterns estimated from the simulated and real data. The similarity was high around these parameters, and at these parameters, the model showed high metastability level. This result suggests that metastable brain dynamics triggered the spatiotemporal patterns. Metastability is a dynamical phenomenon in which the system's state spontaneously cycles between multiple weakly attracting states, like this. Our result suggests that pattern reflected the fixed past that the states repeatedly went through in the metastable brain dynamics. To examine the spatiotemporal pattern in more detail, we applied big step to big data. This dataset contains over 600 subject resting state image data. First, we checked the estimated onset of spatiotemporal patterns. This is a sample onset time series. We calculate the inter-onset interval. This figure shows the probability of the inter-onset intervals. Interestingly, this log-log plot showed a linear relationship, indicating that the inter-onset intervals followed a power law distribution. This result suggests that the emergence of the spatiotemporal pattern was not a Poisson, but a bursty process, which is widely observed in human activity. Then we characterize the spatiotemporal pattern by the similarity with resting state networks. Resting state network is a set of brain regions whose time series are correlated. We downloaded the spatial pattern of resting state network of fMRI from Brain-MAP ICA. For each time in each pattern, we evaluated its similarity with the resting state networks by correlation coefficient. This was repeated by changing time. This figure shows a sample pattern and its correlation coefficient with fMRI resting state networks. The original pattern only showed high correlation coefficient with the default mode network at this time. This means these patterns are correlated with default mode network. But when we decompose the pattern into each frequency band, delta, theta, alpha, beta, and gamma, interesting structure appeared. In the alpha band, default mode and visual networks alternately appeared. This one. In the beta band, default mode and sensory motor networks alternately appeared. This one. To confirm this visual inspection, we conducted a cross-correlogram analysis. We first detected the time point exhibiting large correlation coefficient, this time point, and assigned ones to the time point. We selected two resting state networks, for example, visual and default mode network, and regarded them reference and target. We segmented the target, triggering on the ones in the reference, and averaged the segment across all the subjects. If there is no relationship between reference and target, the cross-correlogram should become flat, like this. On the other hand, if target precedes reference, it should become like this. Therefore, we can infer the timing relationship of the resting state network from the cross-correlogram. These figures show the cross-correlogram. In the alpha band, default mode and visual networks alternately activated with a lag of about 25 milliseconds. In the beta band, sensory motor and default mode networks alternately activated with a lag of about 30 milliseconds. These lags are about half pi radian in the alpha and beta band. This is a schematic representation of the result. Default mode and visual networks alternately activated with a lag of half pi in the alpha band. These are like sine and cosine functions, which are orthogonal. On the other hand, default mode and sensory motor networks alternately activated with a lag of half pi in the beta band. The default mode network is considered a cortical hub. The visual and sensory motor networks are considered to reflect visual and sensory motor information. Therefore, these orthogonal activities may reflect the brain's strategy to achieve both the integration and separation of visual and sensory motor information. When we remember what happened yesterday, such as playing baseball, visual and sensory motor information needs to be transiently integrated without contamination. The spatiotemporal patterns may reflect such process. Furthermore, we for the first time visualize the transition between resting state networks by movies. This is the activity moving from the visual to default mode network. This is the activity moving in the opposite direction from the default mode to visual network. This is a preliminary result showing the velocity vector field estimated from the moving activities. They moved in different ways depending on the directions. This suggests that distinct mechanisms generated the movement in different directions. Finally, I'd like to discuss the relationship to other resting state MEG and EG studies. Many studies have represented resting state brain activity by state conditions. They represent each state lasting about 0.1 second by a static feature, like such as spatial pattern. In contrast, we revealed the brain activity that changed within 0.1 second, like this. Therefore, our study reveals the brain dynamics in much higher temporal resolution. Our works were supported by these findings. I'd like to thank them. Thank you for attention.
Okay, thank you so much for very interesting talk, Takeda-san. So far, we don't have question on the Q&A box. So, but we also accept a question in Japanese as well. So anyway, let me start. Okay, I'm interested in the setting of the step. I mean, how did you choose the window size of for the setting of the step? So, possibly by changing the window size of the spatial temporal pattern, maybe we can find different type of spatial temporal patterns. So, what is optimal size of the this window?
Yeah, actually, step have two hyperparameters, number of pattern and the length of spatial temporal pattern. So you mentioned about this length of spatial temporal pattern. And this is determined by based on generalizability. We divide the data into two data sets and separately estimate spatial temporal pattern with each parameter set. Then select the hyperparameter that exhibit highest correlation coefficient between these data sets.
I see. Okay. So based on the similarity of the spatial temporal patterns between two datasets, you chose the window size.
Yes.
Okay. So actually, that is related to my second question. I'm interested in the sensitivity of the selection of the initial value or something of the model. So I think you said that you randomly set the parameter u at the beginning, then gradually optimize the model. But possibly the difference of the initial value can change the result, final result. So did you see any such kind of instability of the estimation, or did you see that the result is stable, not depending on those initial values?
Yeah, second question. This, in my experiment, this method is robust to initial value because even if I apply this method to different data set, similar result were obtained. So I think this method is very robust to initial setting or even data.
I see. Okay, thanks so much. Okay, we have a question. Well, when the subject transitions to a resting state, how fast is the onset of DMN, default mode network? That is a question.
Thank you for question. In our study, we only use resting state. So to answer your question, this question, we need to apply our methods to task and resting state data. So I think at this time I cannot answer this question.
Okay, then it's time, and let's move to the next speaker. Yeah, Takeda-san, thanks so much again.
Thank you.
Good afternoon, everyone. I'm Yuan Hao. Now I'm a postdoctoral researcher and decan and cooperative researcher at ATL. Today, I'm very happy to present my recent work to this workshop. It's titled Non-Gaussian Brain Source Imaging Using Correntropy-Based Noise Model. Let me start with the background. So brain source imaging is a very useful non-invasive window to investigate the brain dynamics. In source imaging, the non-invasive signals such as EEG or MEG are assumed to be generated from the internal brain activity by a forward model. The internal source activity is linearly transformed by the volume conduction leading to the external observation such as MEG or EEG. We assume the source activity is distributed on endpoints with high spatial resolution, and the brain activity is observed by only M observation channels with low spatial resolution. Here we denote the observation by B, which is M-dimensional vector, and the source activity is S, which is N-dimensional. The volume conduction is called lead field, which is a matrix, and also we have the observation noise on the MEG or EEG recording. Now we can define the source imaging as inverse problem to estimate source activity from MEG or EEG observation. Assume that we have the time series of length t, so we can write each variable as a matrix. The purpose is to estimate S from B and G. The lead field matrix G can be usually obtained from the structural MRI. Because you see, all the recording depend only on non-invasive recording, but now we can know some internal source activity, so the source imaging can help to investigate the internal activity without any invasive recordings. But the fundamental problem for source imaging is ill-posed nature. Usually we will assume so many sources, but we only have a limited number for the observation channels. As a result, we will have N unknowns, but only M equations. The observation matrix can correspond to an infinite number of source solutions. Usually, to solve this kind of ill-posed problem, many studies have proposed different regularization strategies to estimate the source activity, which include minimum norm family or minimum variance beamformer, LORTA, and Bayesian methods. Surprisingly, almost all the source imaging methods can be unified in a Bayesian framework. Next, let me expand the Bayesian framework for the brain source imaging task. First, we usually assume the observation noise has a multi-dimensional Gaussian distribution, where phi denotes the inverse of variance. By the forward model, we can write the likelihood function for the whole observation matrix. Then, to regularize the source activity, we use a Gaussian model as a prior distribution on S, which is conditioned on the hyperparameter A. The inverse covariance for source activity is denoted by FA. If we want to make a full Bayesian inference procedure, we can also assume a hyper prior distribution for the hyperparameter. Then we can write the posterior distribution for the source activity, which is usually very difficult to directly calculate. So there are three ways to solve this problem. First, we can maximize the marginal likelihood for the hyperparameter and then calculate the posterior distribution for S. Then we can also treat the hyperparameter as hidden data through the EM algorithm. Finally, we can estimate the posterior distribution for S and A at the same time by variational inference. The main difference in every source image algorithm is mainly about how to define a prior distribution for the source activity and how to optimize the posterior distribution. However, a very critical problem is about the distribution of noise. MEG and EEG noise can come from system, environment, and physiological artifacts. This means the MEG and EEG noise is non-Gaussian property with high probability. And on the other hand, because the artifacts usually have large value, it means the observation noise is potentially heavy-tailed, which is very harmful for Gaussian noise perception. Then let me introduce the motivation of my research. To solve the non-Gaussian noise for source imaging, I use the concept of coentropy, which is a robust similarity measure. Here, I give the definition of coentropy. It is the expectation of a kernel function between two variables. In practice, we have a finite data set to estimate the value, and the Gaussian kernel function is usually used. Finally, the empirical estimation for coentropy noted by this equation can be first used as a generalized correlation function for signal processing. And also, coentropy is also used as a robust force function for many machine learning tasks. Because coentropy measures the similarity between two variables, we can maximize the coentropy between our prediction and the target, which is called maximum coentropy criteria, or MCC. Here, let me show an example. Here, I write the gradient of the conventional MSE loss and for the MCC loss function. By comparing these two gradients, we can find the MCC loss function as an extra term in the gradient update. This term is plotted in the right figure. When the error is 0, this term will be equal to 1, and when the error is far from 0, this term will decrease to 0 gradually. Therefore, it's kind of reweighting factor according to the error value. So it also explains why MCC is robust to heavy-tailed noise. And this reweighting factor is fully controlled by the kernel bandwidth H. Now we are going to make a noise model from MCC loss function. This loss function can be naturally transformed to be a multiplied form by using another exponential function. Then it becomes a likelihood function. From this likelihood function, we can intuitively derive a noise model, which is simply use another exponential function to the Correntropy similarity measure. However, if I plot the figure of this model, it is not proper for our source imaging task. It is extremely heavy-tailed with no flexibility, because you see, even though the error is far from the origin, the probability density is always 1. We hope a robust noise model can generate to the Gaussian model if the noise is not so heavy-tailed. So, to solve this problem, I proposed a new noise model with the following figures. Here, I introduce another hyperparameter, eta. In this new noise model, H is called tail parameter, which controls the density of the tail. As you can see, when H has a large value, the probability density will be close to 0 when the error is far from the 0. And when H has a finite value, the probability density is not 0. This means this noise model cannot be normalized. And for a small value of H, the tail will be higher, which means more heavy-tailed noise property. On the other hand, eta is called shape parameter, which controls the property of the main distribution peak. A large value of eta corresponds to a narrow peak. So eta is very similar to the inverse variance in Gaussian model. This new noise model has two very important properties. First, the maximum likelihood estimation of this noise model is totally equal to the original MCC loss function. It means it provides a new explanation for co-entry loss function from the viewpoint of noise absorption. And next, it will degenerate to a Gaussian mode with an infinite value on H. This can be easily proved by expanding the Gaussian kernel function. Especially in this case, here you can see the hyperparameter eta will become the inverse variance for this Gaussian model. This means even if the noise is not so heavy-tailed, we can use a large value for H. For example, here, like H is equal to 100, we can approximate a Gaussian model for the noise. And then based on my new noise model, I also propose the following source imaging algorithm. First, I use the individual parameters in my noise model for each observation channel M. It means each channel can have different properties. And then we can write the likelihood function for the whole observation. For prior distribution, I use a hierarchical ARD distribution. Inverse variance, here FA is a diagonal matrix. Each hyperparameter A is assumed with a gamma distribution. And then to estimate the source activity, here I use versional inference, which is also assumed with the mean field approximation. And finally, we maximize ELBO to optimize the posterior distribution. An important problem is how to determine the hyperparameter here, H and eta. Of course, it is very difficult to optimize the hyperparameter during the versional inference. I determine these two parameters before the versional inference. First, I use the observation matrix and lead field to estimate the source activity by a baseline method. Then I compute the reservoir by observation minus reconstruction. This reservoir is used to approximate the observation noise to estimate the hyperparameter. Because, as mentioned before, my noise model cannot be normalized. So here I use score matching to estimate the hyperparameter. After they are estimated by score matching, they will be fixed in the versional inference. Next, I will introduce the performance evaluation for my proposed method. The performance evaluation mainly used a multimodal neural imaging dataset for phase concatenation, which is available at OpenNeural. Here I use the same ARD prior distribution and the same versional inference. The difference is in the noise model. Here I compared Gaussian model, Laplace students T, and my proposed noise model. Here I first make a simulation study. In this simulation, I first use the real-world MEG or EG observation as the lead field to estimate the source activity. This estimation is used as a ground truth in my simulation. The estimation is multiplied by lead field to generate the synthesized MEG or EG. To corrupt the observation, I use the reservoir as a noise repository. The observation noise is randomly sampled from this repository to corrupt the synthesized MEG or EG. And finally, each source imaging algorithm is used to estimate the source activity from the corrupted observation. And the performance is evaluated by aggregate score and RMSE between the estimation and the ground truth. And also, the real-world dataset is directly used for source imaging by different source imaging algorithms. Here are the results for the simulation study. The left figure is MEG-based simulation. The right figure is EG-based simulation. As you can see, across different SNR values, my proposed model always achieves higher aggregate score and lower RMSE. You can also find that the Gaussian model with blue bar always leads to the worst result. Well, if you use a heavy-tailed noise model, it always improves the performance. This means a heavy-tailed noise model is more proper for the observation noise, which is sampled from my noise repository. And next, I directly use a real-world dataset. Here, the source activity is estimated from the trials when the subject received the visual stimulus of facial image. Here we can see the trial average spatial distribution and time series. in spatial distribution, student's T model and my proposed model result in very similar spatial distribution, which are more accurate than the Gaussian model and the Laplace model. And for time series, the results from heavy tilt, including Laplace, student's T, and my proposed model, are very similar and also more accurate than the Gaussian model. This results mean we should choose the heavy tilt noise model for the real-world source imaging task. Of course, we cannot know the ground truth for the real-world dataset. It's hard to tell which algorithm is really better. So I also designed a downstream task for the performance evaluation. I use different source imaging algorithms to estimate the source activities. For the trials, when the subject receives visual and scrambled image stimulus, and then the estimated source activity by different algorithms is classified due to their label. This table shows the classification accuracy for 16 subjects, and clearly the proposed noise model achieved the best performance across different methods for both MEG and EEG. Here, the higher accuracy means the task-related information is more captured by the source imaging algorithm. And finally, I want to answer a question. Is really the proposed noise model better for the MEG or EEG observation noise? Because we cannot know the true observation noise, here I use this reservoir to examine each noise model. Specifically, I estimate the distribution parameter for each model to fit the distribution of this reservoir. The figure shows the noise histogram and fitting results. You can see the Gaussian, Laplace, and the student's T models deviate from the empirical histogram. But in contrast, my proposed noise model leads to much better fitting. This means, at least in the noise repository, the proposed noise model is really better to fit the observation noise. Let me summarize my talk. So in my study, I first proposed a Correntropy-Based noise model. The maximum likelihood estimation of this model is totally equal to the NCC loss function. It is heavy-tailed, non-normalized, and will become Gaussian model in a special case. Next, I also propose a source imaging algorithm using this new noise model. It is optimized by versional inference and also achieves better performance on both simulation and real-world dataset. For future work, first we can naturally combine this new noise model with other prior distributions for further improvements. And next, I'm also considering a model-free approach to handle the MEG or EEG noise for Bayesian source imaging, because the specific model always leads to some limited flexibility. This is all my presentation. Thank you very much for your attention.
Okay, thanks so much for very interesting talk, Lisa. Okay, so far we don't have a question in the Q&A box. So, okay, let me start from a question. Okay, so it's actually very interesting that by considering heavy tail noise, we can somehow improve the source reconstruction performance. But what do you think, what is the main source of those heavy tail noise actually? So is it related to simply environmental noise, or is it somehow related to brain activities?
So actually, according to my personal experience, I think the heavy tail mainly comes from physiological artifacts, because when you blink or you move your eye, so at least the EEG signal will have a very large fluctuation in the waveform. So, and also, I'm not sure whether the system and environment will give very large value, but at least the physiological ones usually lead to the heavy tailed property.
But basically, you think that those noises are because of those artifacts rather than brain activities?
Yes, and also for MEG signal, if you make some movement of metal materials, it will also have a very obvious impact. So I think it's also kind of source for the heavy-tailed noise.
I see. Thank you. My next question is that I think your main target is the source reconstruction from EEG and image signals. But do you think that this framework, I mean, this noise model can be used to other measurement modalities like fMRI or calcium imaging or spiking data or those type of modalities?
I think for those signals like fMRI with pretty low temperature resolution, I think it will not be so heavy-tailed like this noise model. But anyway, I think it will be also non-Gaussian. So I think maybe using another non-Gaussian model will be more proper.
It depends on what kind of
Yes, I think the real source of the noise is really important to choose a proper noise model.
I see. Okay, thanks so much. It's already the time to finish this session. So yeah, thanks so much, Wei-san, again.